afternoon din. So, ngayong hapon ba i-share ko yung results ng thesis ko, which is actually entitled Species Limits in Purple-Throated Songbirds, Buzzing Flower Peckers, and Philippine Dragons. So, yun. Philippines. Philippines is an archipelago. And being an archipelago, we have high biodiversity. Specifically, high avifaunal diversity. In Kennedy's book in 2000, he cited that 576 are already recognized species, wherein 172 are Philippine endemics. With this high biodiversity, we need to have a re-evaluation of the taxonomic status of our Philippine birds. This is because we have two types of taxonomists. We have longpers and splitters. Longpers meaning they have a tendency to put to have one species with many subspecies, while splitters tend to elevate subspecies to full species, even though the differences between populations are quite small. So to re-evaluate the taxonomic status of species, we have to identify species limits. And species limits is defined as the process of identifying biological diversity in the species level. We can identify species limits in various ways depending on the species concept that you believe in. So, for my thesis, I follow the biological species concept, which is defined as reproductive isolation, or that a species is a member of a population which interbreed in nature without gene transfer from one population to another. Yeah. So, as I said earlier, my thesis made use of the Tobias criteria, and Tobias criteria follows the biological species concept. So, this. The quantitative criteria for species delimitation by Joseph Tobias and his other colleagues was published last 2010. And he published this to establish a standard criteria that could be applied to different bird taxa. So, para pare-pareho na yung means natin ng species delimitation. In the Tobias criteria, there are 5 characters that he used. First is morphometrics or biometrics, followed by acoustics, then plumage and bare parts coloration, ecology and behavior, and lastly, geographical relationships. First, I would like to introduce to you Cohen's d. Cohen's d is the statistical tool that is used in the Tobias criteria. It is an effect size that measures differences between two populations. Sa pag-compute ng Cohen's d, we only use two numbers, the mean and the standard deviation following this equation. So, bakit ito mahalaga? Because I will explain later. So, ayan, ito. So, first, na taxonomic character na ginamit sa Tobias criteria is the biometrics. Cohen's d is used to measure the differences between the bill length, wing chord, tarsus length, and tail length between two populations na pinagko-compare. So, if the Cohen's d, the resulting Cohen's d is between 0.2 to 2, it is considered as a minor difference and is given a score of 1. As follows, with an effect size of 2 to 5, medium, 5 to 10, major, and greater than 10 as exceptional. Next is the acoustics, which I did not use in my thesis because it is quite hard to get samples ng bird songs na pwedeng gamitin. Since ang kailangan ko ay from multiple subspecies, it is quite hard kahit hanapin siya sa Sinocanto kasi ang usual lang na makikita ay yung nominal subspecies. So for acoustics, ang ginagamit dito ay yung temporal and spectral characters like total number of notes, duration of song, the pace or the number of notes in a duration, the maximum and minimum frequency, the bandwidth, and the peak frequency of the song. Next is the plumage and bare parts coloration. So, for a minor change, as we can see here, there is just a slightly difference in the wash or yung suffusion ng color dun sa sample. For example, yung isang bird ay yellow while the other one is slightly lighter in color, or mas marami yung yellow parts ng isang sample. For sample to have a medium difference, there should be a distinctly different tone or shade to its part. As seen in the figure, yung nasa top ay color blue, while yung blue yung throat, while yung nasa baba ay medyo purple na siya. So, next is the major. Kitang-kita natin dito na contrastingly different na yung colors ng ostrich. Yung isa ay blue at yung isa ay orange. This last, exceptional, dito dapat ibang-iba na talaga yung itsura nung dalawang sample mo. For example, it could be a major difference in color, or it could be a difference in design. If your sample has spots on it while the other one has striations. Next is ecology and behavior. I wasn't able to use this character in my thesis because it is quite hard, because I need to go on field to observe this place or this behavior. So, a non-overlapping differences in foraging or breeding habitat, adaptations, and other adaptations is considered as a minor change, while non-overlapping differences in courtship display is considered as a medium change. And lastly, the geographical relationship. A score of 1 is given if the hybrid zone is quite broad. A score of 2 is given if the hybrid zone is narrow. And a score of 3 if the population is parapattern. And no score is given if the population is allopatric. So here is the summary of how many values per character can be used in the computation for the cumulative score. So for the biometrics, only 2 will be considered, the greatest increase and the greatest decrease. For acoustic, the 2 strongest temporal and spectral character. For plumage and bare parts coloration, they're the top 3. And for ecological and behavior, only 1. So, to merit a split between 2 compared populations, you must have a cumulative score of 7 or greater than 7. To give you a sample of a study that made use of the Tobias criteria, here is a paper of N.J. Collard entitled Species Limits in Some Philippine Birds, including the greater flameback Rhizocolaptes lucidus. This was published in Forktail last 2011. They established some new species and verified the full species status of some birds. For example, the Philippine collared dove, the Philippine green pigeon, Boer green pigeon, and other Philippine bird species. With that, let me introduce to you the 3 bird species that I tackled in my thesis. First is the purple-throated sunbirds or Leptocomas sperata. They are formerly named as Nectarinia sperata. They are native in the Philippines, and they can also be found in other countries in the Southeast Asia. They are dimorphic and belongs to the family Nectarinidae, order Cassariformes. Only 4 subspecies are found here in the Philippines: Leptocomas sperata henke, sperata, Trochilus, and Juliae. To give you a more specific view of the subspecies, here is the overall distribution of the Leptocomas sperata. Well, here is this in the Philippines. In the northern Luzon, we have Leptocomas sperata henke. The rest of Luzon, Leptocomas sperata sperata. In Palawan, in Visayan area, and some parts of Mindanao, we have Leptocomas sperata Trochilus, and in the rest of Mindanao, Leptocomas sperata Juliae. Next is the buzzing flower peckers, Dixeum hypoleucum. It is endemic in the Philippines and is monomorphic. It belongs to the family Dicaeidae, order Cassariformes, and it has 5 subspecies. So, this is the distribution of Dixeum hypoleucum. I want you to take note of the 2 races of Dixeum hypoleucum. We have black and white race and the olive green race. In the northern race is the olive green race. First is the Dixeum hypoleucum cagayanense, which is found on the northern Luzon, and Dixeum hypoleucum obscurum, the rest in southern Luzon. The black and white race is found in the Visayan and Mindanao areas. For the Visayan area and some parts of Mindanao, we have Dixeum hypoleucum pontifex, and for the islands of Tawi-Tawi and Basilan, we have Dixeum hypoleucum and Dixeum hypoleucum mindanense, which is limited to Zamboanga Peninsula. Lastly is the Philippine trogons, Harpactis ardens. These are endemic species and are dimorphic. They belong to the family Trogonidae, order Trogoniformes, and there are 5 subspecies of the Philippine trogons. First is Harpactis ardens herberti, found in the northern Luzon, and Harpactis ardens luzoniensis, found in the rest of Luzon except Polillo. And this is Harpactis ardens minor, which is only found in Polillo. And unfortunately, I can't find any picture of the female of the male minor. And the Visayan area, we have Harpactis ardens linnae, and the rest of Mindanao, Harpactis ardens ardens. So for my thesis, I gathered my samples from the Museum of Natural History, here, UPLB, the National Museum of the Philippines, and I've also requested morphometric data from the Museum of Zoology, University of Michigan, and the Yale Peabody Museum of Yale University. In totality, I measured 515 male museum skin specimens. And here is their breakdown. Take note that I've only measured male specimens because in the avian world, the mating is driven by female choice. So male birds are the one who adapt. So they could attract the females. So here is what I've done. I've measured the wing chord, the bill length, tail length, and the tarsus length of this museum skins, and compared the plumage and bare parts coloration between series of samples. Then I made a matrix wherein I made sure that one subspecies is compared to all other subspecies. Taking extra careful that I have been able to compare each subspecies with each other. So for my results, First, for the Leptocoma spirata, I want you to notice every comparison between the subspecies Leptocoma spirata henke. In all comparison between Leptocoma spirata henke, the cumulative score reached 7 and higher, meaning it warrants split from the other subspecies. Also, I want you to notice the scores for Leptocoma spirata juliae. However, for the last comparison between Leptocoma spirata trochilus and juliae, it did not reach the score of 7, but it is still high at 6. And because at other comparison, it reached the cumulative score of 7, I also proposed a split for this subspecies. And how did I arrive with this? On the top left, we have Leptocoma spirata henke. Leptocoma spirata henke is different from the other subspecies of Leptocoma because it has an all-black back and wings, which is different from the other subspecies which has a reddish color. For the Leptocoma spirata juliae, it can be seen that their breast coloration is very different. The other three have orange-red, while it has yellow coloration with an orange stripe in the middle. So this warrants a major difference between the other subspecies. For the morphometric measurements, the main observation here is that the tail of the juliae is significantly smaller compared to spirata, while all the others remain insignificantly different. So for the results, I am proposing the splitting of Leptocoma henke and Leptocoma juliae, leaving 3 species for Leptocoma: Leptocoma spirata, Leptocoma henke, and juliae. While spirata have 2 subspecies: Leptocoma spirata and Leptocoma spirata trochilus. Mentioned earlier, there are two races within Diceium hypolupum. And as you see here, when comparison were made between the 2 races, the cumulative scores reached above 7. And this, for the southern race, we have the black and white, and the northern race, we have the olive green race. So, seeing these 2 pictures, we can easily see the magnitude of the differences between the coloration of the 2 races. These differences warrants the high scoring, then therefore resulting to the high cumulative score I have presented before. Also, for the morphometric measurements, I have observed that for the tail length, the tail of the Diceium hypolupum is larger compared to that of the obscurum, which is a part of the northern race. Also, the tarsus length of there is also a difference between the tarsus length of the subspecies hypolupum and the Mindanense. Can be seen that Mindanense has a shorter tarsus compared to that of hypolupum. And with that, I suggest the following splits: that the 2 races be split from each other, forming the 2 taxa Diceium hypolupum and Diceium obscurum, retaining the other while Diceum hypoleucum have the subspecies Diceum hypoleucum hypoleucum and Diceum hypoleucum indanense and Diceum hypoleucum pontifex. And the other taxa, Diceum obscurum, having the subspecies Diceum obscurum obscurum and Diceum obscurum cagayanense. Lastly, for the Harpactes ardens, none of the comparison resulted to a cumulative score of greater than 7. This is because there's very few or minute differences between each subspecies. They all look quite the same as can be seen there. And I also encountered a problem wherein the bright red coloration of the breast of the trogon disappears after a long time. So I wasn't able to use the breast part for the bare parts coloration, but I have to make use of the other parts such as the back and the wings, wherein I wasn't able to observe any major differences. So for the morphometrics, I have observed that for the wing, between ardens and minor has a shorter wing chord, and between herberti and minor has a shorter wing chord. The same is true between linnae and minor. Further, I also observed that between linnae and minor has a shorter bill, and between linnae and herberti has a shorter bill. This observation further proves that the island endemic subspecies Harpactes ardens minor is indeed the smallest subspecies within the Harpactes group. Given that, I propose that there will be no split, or that the taxonomic status of Philippine trogons be retained. So why is it important that we reevaluate the taxonomic status of Philippine birds? First, because of island endemicity and biodiversity. With a change in the taxonomic status, we can increase or decrease the biodiversity of here in the Philippines. Also, as I've shown earlier, with my proposed split, we could have island endemic species that is new to the country. Also, taxonomic changes lead to changes in conservation status. For now, the 3 species are considered as least concern. But if my proposed taxonomic changes be further validated, the area of occupancy of these species will be halved or will be decreased. Therefore, this will have an effect on the conservation status that the species have. And knowing the conservation status of these species, we will be able to provide proper conservation actions. So we can protect this newly elevated species, and we can prevent further decrease of their population. So yes, hope you learned something. Thank you.